This method can help answer key questions in livestock entomology, such as what stable flies require for development. The main advantage of this technique is that it is simple. The medium is composed of only 4 ingredients.
Propagating Stoimoxis calcitrans begins with collecting eggs from a stock of flies. This involves preparing an egging cup. Dip the tip of a cloth into a 250 milliliter jar filled with 40 degrees Celsius water. Then wrap the cloth around the outside of the jar and secure the cloth with a rubber band. Fold the loose end of the cloth over the top of the jar to limit access to the water. Now place this cup into a cage of 8 to 10-day-old flies. After 2 hours, the gravid females will oviposit enough eggs for the next generation. Typically, 2,500 flies will produce 8 to 40,000 eggs in 2 hours. Next, using water, rinse the eggs off the egging cloth into a small, empty dishpan. Theoretically, eggs can survive in water for 8 to 12 hours. However, it is best to limit the time to 5 hours or less. To rear larvae from the eggs, make up fresh fly medium in a 10-liter plastic dishpan. Then make a shallow furrow in the medium along the length of the dishpan for the eggs. To transfer the eggs, make a specialized egg pipette by inserting a mesh screen into a pipette. Simply cut the pipette at the desired location and glue a number 100 mesh screen into the